Ever since the beginning of time, people have been intrigued by the mystery of life. Where do we come from? Why are we the way we are? In a revolutionary discovery, James Watson, Francis Crick, Maurice Wilkins, and Rosalind Franklin answer this age-old question. The reaction throughout the scientific community rang loud, reforming and improving modern biological and medical sciences. The discovery of the structure of DNA has led to a better understanding of DNA and has created a vaster understanding of genetic diseases and knowledge of how life recreates. A basic awareness of genes in DNA was not new to Watson and Crick, as Austrian scientist Gregor Mendel had already discovered the gene, and in 1865 published his findings in a monograph called Experiments on Plant Hybrids, Revolutionizing the Field of Genetics. After Mendel's work, the field of genetics started becoming more accepted. In 1869, scientist Friedrich Miescher was working in his lab in Germany when he accidentally discovered DNA by putting pus in a weak alkaline concentrate, leaving only a reformed state of DNA. This he called nuclei. Then, in 1944, Oswald Avery, McLean McCarty, and Colin MacLeod, working at the Rockefeller University in New York, showed through extensive experiments that DNA was the genetic material, and not proteins as many had believed.
And purified out the protein, the DNA, and the RNA. Which one of these components is able to transfer the virulence from the avirulent bugs to the virulent bugs? To everyone's shock and amazement, it was the DNA. Okay, so I take a molecule now, which no one had cared about since it was discovered by Friedrich Miescher, right? I've now taken that from being this, and all of a sudden with the experiment of Avery, McCarthy, and MacLeod, it moves to center stage in the study of genetics.
Their findings led to the advancement of research of DNA in many labs around the world. In order to understand what DNA was, scientists in the 1940s tried to create models of DNA. One of the leading scientists in the field at the time, Austrian biochemist Erwin Chargaff, reformed the study of DNA when he noticed a pattern in the amounts of the four main bases: adenine, thymine, guanine, and cytosine. This discovery later helped with the final conclusion of Watson and Crick.
In 1951, J.T. Randall, the current conductor of the physics department at King's College, recruited Rosalind Franklin as an X-ray crystallographer. In September of 1951, Franklin discovered that there were actually two forms of DNA, dry form A and wet form B. In the same year, while working at Cavendish Laboratory, James Watson, an American scientist, and Francis Crick, an English scientist, became partners in the race to find the structure of DNA. Using many ideas from American researcher Linus Pauling, they decided to go straight to building a model. Their first model, a triple helical replica of DNA, was a misconception. The university reacted by discouraging Watson and Crick from working any longer on DNA. As 1952 dawned, Rosalind Franklin and fellow researcher Raymond Gosling found a dead end in crystalline X-ray form A. Franklin continued working day and night to perfect the B form, and on her 51st X-ray photo, she got it right. It was mid-1952, and Watson and Crick were extremely discouraged. When Linus Pauling's triple helix paper was published, they knew he was wrong, so they began their research again at Cavendish Laboratory, getting straight to work with their new colleague Peter Pauling, the son of Linus Pauling.
By 1953, Franklin had no idea that the race for the double helix had started. However, she looked again at photo 51 and concluded that both forms A and B were made up of helical chains, so she reacted by continuing further research to find the structure. Watson and Crick had actually discovered the structure of DNA on February 28, 1953. They quickly shared their findings at a pub in Cambridge known as the Eagle. On April 25th, Watson and Crick published their revolutionary discoveries in a one-page paper, A Structure for Deoxyribose Nucleic Acid, in the British scientific weekly Nature.
Did her photograph, or whatever you call it, contribute to it?
Oh, sure, because when I saw it, had to be a helix.
This monumental discovery was the end of the race for life. Unfortunately for them, their victory was tainted. They'd stolen the work of fellow researcher Franklin and passed it as their own. In 1962, the Nobel Foundation awarded Watson, Crick, and Wilkins the Nobel Prize for Physiology and Medicine, reacting to their discovery. Sadly, Franklin, whose work was stolen by Watson and Crick, died in 1958 and could not be nominated for a Nobel Prize. To this day, she is still known as the Dark Lady of DNA.
It was impossible. You only can give a Nobel Prize to 3 people, so there have been 4 of us. So the Swedes could have given 2 prizes.
Her work, along with the work of all the DNA scientists, has led to many reforms in science. One of the major effects was our instant knowledge of how life recreates itself with some regularity. Each strand of DNA holds a certain gene. All of these genes together make up who we are and how we grow. Because of this discovery, we now know the structure of DNA is rather like a spiral staircase. When the gene reproduces, it goes through a process of mitosis. This is when the gene divides into 2 identical strands of DNA. Thus, the physical characteristics of a child are like the parent. Starting in 1973, scientists began to genetically engineer plants to make them bigger, seedless, or even a different color. A similar process has been used to create clones of plants and animals. The first cloned mammal was a sheep, Dolly, cloned in 1996. The Roslin Institute in Scotland created Dolly from an adult cell. Dolly was the only living clone out of the many attempted. She died in 2003. Now, because of her, scientists understand how to clone. Also, the cloning process used to make Dolly has led to a reform in cloning.
Another branch of DNA research is DNA splicing. There is talk that by using this method, soon humans may be able to shape children before they are born. In order to discover and understand the DNA of living things, scientists use a process called DNA sequencing. This process has been used as a basis for many projects and scientific breakthroughs since 1976, when Alan Maxam and Walter Gilbert used chemical modification to sequence DNA. Today, we have used it to sequence the human genome and in DNA modification. This project was completed in 2003. Humans are now reading the genome.
Having the complete human genome sequence has in turn led to things like the Human Genome Diversity Project.
Once they understand it, scientists will know humans at the most basic level. Once they know the human genome, the scientists can react by changing us, and doctors can cure diseases more easily. In recent years, the fact that each person has their own unique DNA has been used in the enforcement of criminal justice. Like many cases around the world, in 1999, the New York Police Department used DNA to convict a man for a chain of sexual assaults and robberies. Also, because of the discovery of the structure of DNA, the science community is now investigating ways to prevent harmful genetic diseases such as Alzheimer's and Parkinson's, and has discovered that paranoid schizophrenia can be prevented with pills, therapy, or special lifestyles. Mankind has always been intrigued by where we come from. Because of the work of James Watson, Francis Crick, Maurice Wilkins, and Rosalind Franklin, we are now beginning to understand. We know the reason we can reproduce and pass on our genes. Although Franklin may have reacted poorly to Watson and Crick stealing her work, they still had a large impact on the world. Watson and Crick created a revolution by finding the structure of DNA, but they could not have found it without the work of the other scientists. The Nobel Prize Watson, Crick, and Wilkins received is a symbol of their accomplishment. The revolutionary discovery of the structure of DNA has reformed science so much more than the researchers could ever have imagined.
So today we're going to talk about DNA, and we're going to go into the structure and the function of DNA. But before we do that, we want to talk about why DNA is so awesome. So let me give you a not so real world example. If you've ever seen the movie Jurassic Park, you know it has the storyline where some scientists find a mosquito trapped in amber. Now, the idea is this mosquito had once fed on a dinosaur, so it had dinosaur blood inside it. The idea being that it had a molecule then that was in this blood known as DNA, and they used the DNA to clone and create dinosaurs. Now, there are some unrealistic things in this, but the idea of cloning is not unrealistic because actually it can and does occur. Say, for example, you wanted to clone an iguana. I don't know why I picked iguana. I think I just like the name iguana. Well, if you wanted to clone an iguana, you would need to have a reptile egg cell, and you would need to take the DNA out of that reptile egg cell. And then you would need to put the DNA of the iguana into that reptile egg cell. So basically, you took the DNA out, you made it kind of like an empty slate, and you put the organism you wanted to clone, the iguana, you put its DNA inside that egg cell. That egg cell is basically now programmed to develop into that iguana, that organism. It would need to be implanted somewhere so that it can develop. Now, keep in mind that it still would hatch into a baby iguana, but over time, that iguana would develop, and there'd be some environmental conditions that might make it look slightly different than the original because the environment can affect how the organism will look and also how genes function. But the iguana would be a clone, an identical copy, because the genetic material, that DNA, would be the same. There are a lot of ethical debates with cloning, or really any biology topic that involves DNA, because DNA is the most important molecule with life. It controls all of your traits. Your cells can't function without it. How tall you are, what color your eyes are, what color your hair is, or even if you're at risk for certain diseases like heart disease or cancer, some of that can be found in the DNA. Your DNA is just incredibly important. One thing, too, that students sometimes don't understand is that all of the cells in your body, every single cell, has your entire DNA code, each of them. So, if you've ever watched those shows, CSI or Law and Order, you know how the criminal can leave behind a hair sample or a skin sample or a blood sample, and they can link that and find the criminal sometimes if they get a good sample. The idea is even leaving behind a hair that has hair follicle cell, you can get the entire DNA code because every cell has all of your DNA. One thing to point out, even though all of your cells all contain your entire DNA code, that DNA is not turned on all the time in all of your cells. Let me give you an example. A skin cell is not producing digestive enzymes. That would be nasty. The digestive enzyme, even though it's coded for in the DNA, and your skin cells, like all cells, have all of your DNA, that is turned off in your skin, which is a good thing. Well, in maybe a stomach cell, that would be turned on, be activated. We call that gene regulation, the ability to turn genes on and off. So let's talk about the structure of DNA. First of all, DNA is a type of nucleic acid. And if you remember the biomolecules, remember how the biomolecules included carbs, lipids, proteins, and nucleic acids. Nucleic acids are the type of biomolecule that DNA falls under. And like all biomolecules, there are building blocks called monomers. The building block of a nucleic acid is a nucleotide. Okay, so I know I'm kind of bombarding you with vocabulary, but it's really important to understand the parts of a nucleotide because otherwise you don't know what DNA is made of. And if you don't understand what DNA is made of, you won't understand how it functions. And if you don't understand how it functions, well, that's going to be really difficult when we really start understanding how we work. Before I get into the three parts of a nucleotide, I do want to point out the name of DNA. So DNA stands for deoxyribonucleic acid. That's what the Na part is, nucleic acid. And that D, that deoxyribo, that's for deoxyribose, which is a sugar. So nucleotides have 3 parts, and one of them is a sugar called deoxyribose. The next thing is a phosphate. The phosphate is involved with the sugar in helping hold the DNA structure together. We sometimes call DNA as having a sugar phosphate backbone. But the most important part of the nucleotide is the base, because the bases actually code for traits. So as far as the bases go, again, those are the important parts that control your traits. They really directly code for your traits. There are 4 bases in DNA. And a lot of times they'll just use the initials, A, T, G, and C. The A is for adenine, the T is for thymine, the G is for guanine, and the C is for cytosine. There's a really nice little saying that can help you remember which one goes with which, because these bases actually pair in a correct pattern. And if you don't have them paired correctly, or maybe they get mismatched, that's actually what we call a mutation, which we'll get to a little bit later. But here's the little way you can remember it. Apples on the tree. That tells you that A for apples, T for tree, A and T always go together. And then the other little verse is cars in the garage. That can help you remember that the base C always goes with the base G. And so you kind of know how they match. And DNA pairs this way. DNA has 2 strands. So there's nucleotides running up one side, and there's nucleotides running up the other side. The bases are what pair in the middle. They're usually held together by hydrogen bonds. The DNA is also twisted in something we call a double helix shape. And so as the strands of the DNA is twisted around, the bases should be all in the center. The sugar and phosphate make up the sides. So let's review what we talked about today. We talked about the importance of DNA, that it's found in the nucleus of cells, that all of your cells, every one of them, they contain all of your DNA, although the DNA is not always turned on completely in all the cells. It depends on what the cell's function is, whether it's going to be turned on or off. That's called gene regulation. We talked about DNA is a type of nucleic acid, and that nucleic acids, which is a biomolecule, are made of nucleotides. And nucleotides contain 3 parts, a sugar, a phosphate, and a base. It's the base that codes for your traits. And we talked about which bases match. You can help remember that apples in the tree, cars in the garage, to give you a little nice way to remember it. Remember, we really have to understand DNA in order to understand how our own bodies function. And when we get into genetics, we'll talk about how that DNA actually works and how it really codes for traits. That's it for the Amoeba Sisters, and we remind you to stay curious.